We are now ready to answer the question, are there fragile regions in the human genome? Here's a theorem. Yes. And here's the proof. If the random breakage model is correct, then n rearrangements applied to circular chromosome will produce approximately 2n synteny blocks. And therefore, since there are 280 human mouse synteny blocks, there must have been approximately 280 over 2, which is 140 two breaks on the human mouse evolutionary path. But we have just learned from the two-break distance theorem that there are at least 245 two breaks on this path. My next question is 245 equal to 140? It turns out that 245 is much larger than 140, even if you apply rigorous statistic allowing for some differences between these two numbers, which means that we arrive at a contradiction, implying that one of our assumptions in the course of proving this theorem is incorrect. But which one? Well, there is not much choice because we have made only one assumption. Here it is. If the random breakage model is correct, and since based on this assumption, we arrived to a contradiction, it means that our assumption was incorrect, which implies that random breakage model failed. Well, we demonstrated that random breakage model is incorrect, but of course we have not provided you with the location of fragile regions in the human genome, and it is an example of non-constructive proof, when we prove that something is wrong but cannot provide a certificate showing where the fragile regions are located. But if the random breakage model is incorrect, how would you explain the remarkable fit between the observed length of synteny blocks and the exponential distribution? Well, why have biologists embraced the random breakage model? Actually, it was a logical fallacy. RBM, random breakage model, is not the only model that complies with the exponential distribution test. So it is correct to claim that random breakage model implies exponential distribution, but it is absolutely incorrect to claim that exponential distribution implies a random breakage model. So why was random breakage model refuted? Because it doesn't comply with the observed breakpoint reuse. And the question is there a model that complies with both the exponential distribution and the breakpoint reuse phenomena? And it turned out that so-called fragile breakage model indeed explain both phenomena. According to the fragile breakage model, our genomes represent a mosaic of fragile regions with high propensity for rearrangements and solid regions that hardly ever broken by rearrangements. And fragile regions, which means regions between consecutive synteny blocks, are short, accounting, let's say in the case of human genome, for less than 5% of our genomes. Now, the question of course arises how FBM, fragile breakage model, explain both exponential distribution and the rearrangement hotspot. Well, a small number of short fragile regions explain rearrangement hotspot because there are very few regions where rearrangements actually can happen. Therefore, they are bound to be repeated in the same places. Also, if fragile regions are somewhat randomly distributed throughout the genome, then the synteny blocks lengths follow exactly the same exponential distribution as in the case of the random breakage model. And therefore, a fragile breakage model explains both exponential distribution and breakpoint reuse and should be used as a substitute for the random breakage model. But maybe a fragile breakage model uses the same logical fallacies that the random breakage model uses, because maybe there is a test that both random breakage model and fragile breakage model fail. It turned out very recently that indeed there is such a test. And this test became possible because many genomes have been sequenced in recent years, and scientists were able to analyze breakpoints not only between two genomes, human and mouse, the first sequenced genome, but between multiply genomes. And they proposed so-called multiply breakpoint reuse test, which analyzes breakpoints across multiply genomes. Unfortunately, we don't have time to go into the details of the multiply breakpoint reuse test, but I can only tell you what follows from studies that analyze this test. This recent study revealed evidence for birth and death of the fragile regions, which means that fragile regions exist, but they are not static. They move to different locations in human and mouse genomes, and the positions of the fragile regions in human genome are different than the positions of the fragile regions in mouse genomes. And this discovery resulted in the turnover fragile breakage model, which allows for changes in the location of fragile regions, and this new turnover fragile breakage model actually complies with a new multiply breakpoint reuse test. Moreover, it actually points to the location of the currently fragile regions, the questions that we were not able to answer when we were proving the last theorem from the last section. So a turnover fragile breakage model actually complies with all three tests. Does it mean that it's absolutely final model for chromosomal evolution? Of course not. There is a possibility that future studies will reveal new tests and will lead to further development of our views of chromosome evolution. And with turnover fragile breakage model developed, there are still questions that remains to be answered. Where are the fragile regions located, and what causes fragility? And availability of multiplied genomes will help us eventually to answer this question and to figure out what are the currently active fragile regions in the human genome. What will be maybe the next reversals that happen in the human genome, let's say in the next million years. And now, after we covered different algorithmic aspects of genome rearrangement, the time has come to answer one of the first questions that appeared in this lesson, is how to generate synteny blocks starting from long genomes.